All right, buckle up, everybody, because today we are diving deep into a paper that's making some serious waves in the world of genetics.
Oh, yeah. This one is a doozy.
It's called How Life Changes Itself. The Read-Write RW Genome by James A. Shapiro.
Catchy title.
Right. And it's a pretty bold statement, too.
It is. We've always kind of thought of the genome as like this fixed blueprint, right?
Yeah.
This instruction manual for building an organism that you can't mess with. But Shapiro's basically saying, hold up, not so fast.
So is he saying that our cells can, like, edit their own DNA? That sounds like, I don't know, some sci-fi movie stuff.
It's actually happening right now in every cell in your body. I mean, the traditional view is that evolution is all about random mutations and then natural selection picks the winners. But Shapiro is arguing that cells have an entire toolkit for actually restructuring their genomes. It's a process he calls natural genetic engineering.
NGE. Sounds intense.
It is.
So are you saying it's not just about random changes? Like, cells are actually in control, making intentional edits to their DNA?
Exactly. Think of it this way. The old view is like a fixed blueprint, right? But Shapiro is saying it's more like a complex document that the cell can actually revise and update as needed.
Okay, so my mind is officially blown. So how does this NGE thing actually work? Like, what tools are we talking about here?
Well, cells have a pretty incredible array of molecular tools at their disposal. For example, they've got these things called endonucleases, which act like molecular scissors that can cut DNA at very specific locations.
Wow.
Then you've got ligases, which are kind of like molecular glue that can join DNA fragments together.
Okay.
And get this, they even have reverse transcriptases.
Reverse transcriptases, what are those?
Those are enzymes that can copy RNA back into DNA.
Wait, back up a sec. Cells can copy RNA back into DNA. I thought that was a one-way street, like rewriting the source code, right?
It is. It's groundbreaking, right? So we're talking about cells having this whole toolbox that's incredibly similar to what we use in genetic engineering labs, but they've been doing it naturally for like billions of years.
That is amazing.
Right.
So we've got these like cellular editors running around, cutting and pasting DNA. But what's the point? Like, why would cells want to change their own genomes in the first place?
Well, think about it. The environment is constantly changing, right?
Yeah.
Organisms need to be able to respond to those changes to thrive.
Yeah, that makes sense.
So NGE gives them a way to do that actively rather than just waiting for random mutations to come along.
So it's like evolution has a fast forward button.
Yeah.
Instead of just waiting for random changes, cells can use NGE to speed up the process, adapt more quickly.
Exactly. And the implications of this are huge. It means that evolution might not be as slow and gradual as we once thought, and it suggests that cells play a much more active role in shaping their own evolutionary destinies.
This is fascinating, but it also brings up a ton of questions. Like, if cells are so good at manipulating their own DNA, why do we still see harmful mutations? Shouldn't NGE be able to just fix those?
That's a great question, and it's something that Shapiro addresses in his paper. NGE isn't a perfect system. Sometimes those changes can backfire and actually be detrimental.
Oh, so it can actually cause problems too.
Yeah, and the cellular machinery that regulates NGE can sometimes go awry, leading to unintended consequences.
So it's like any powerful tool, right?
Yeah.
It can be used for good or bad. And sometimes things go wrong, even with the best intentions.
Absolutely. It's important to remember that evolution isn't about achieving perfection. It's about finding solutions that work well enough in a given environment. And NGE is just one part of a complex, dynamic system.
That makes sense. So it's not like NGE is deliberately trying to create harmful mutations. It's just that sometimes those changes have unintended consequences.
Right. And to fully grasp the implications of this, we need to dive into how these NGE tools work on different timescales.
Timescales, you mean like how quickly these changes are happening?
Exactly. Shapiro breaks down genome changes into these categories based on how fast they occur. Some happen within a single cell cycle, others over multiple generations, and some even over vast evolutionary time.
So we've got these like layers of complexity, from quick edits during cell division to those long-lasting changes that get passed down through generations. It's like the genome's constantly evolving, but at multiple speeds simultaneously.
And that's just within a single cell.
Yeah.
When you scale that up to populations and ecosystems, the complexity, it just explodes. But to really understand how this impacts evolution, we need to look at some specific examples.
Okay, so where do we start? What's like one of the most dramatic ways NGE is shaking things up?
Let's talk about those molecular scissors, the endonucleases.
Okay.
They're not just cutting DNA randomly. They're often targeting specific sequences, sometimes even introducing intentional breaks to trigger a response.
Intentional breaks. That sounds risky. Why would a cell want to damage its own DNA?
It's not about damage. It's about controlled change. Sometimes a cell needs to shuffle things around, rearrange its genetic deck of cards, so to speak.
Right.
And sometimes a break in the DNA can trigger repair mechanisms that actually introduce new variations.
So it's like a controlled explosion.
Yeah.
Breaking things down to build them back up in a new way.
That's a great analogy. And it turns out that cells have an incredible capacity for repairing these breaks, often using those legacies we talked about earlier to paste things back together.
This is mind-blowing. It's like cells are playing this high-stakes game of genetic Jenga, pulling out pieces and putting them back in different places. But how do they know which pieces to move? Is there some kind of master plan?
That's where things get even more fascinating. Shapiro points to this phenomenon called untargeted mutagenesis. It really challenges the idea that mutations are purely random.
Untargeted mutagenesis. So there's a purpose behind some of these mutations.
It seems that way. He cites these fascinating experiments from the 1950s by Gene Weigel, where bacteria, when they're exposed to UV radiation, they actually increase the mutation rate of viruses that they infect.
Really?
And this wasn't just random damage, it seemed like a deliberate response.
Almost like the bacteria were saying, hey, times are tough, let's shake things up and see what works.
Exactly. This completely shatters the idea of cells being passive victims of random mutations. It suggests that they have ways of sensing danger and responding by actively exploring new genetic possibilities rather than just waiting for mutations to happen by chance.
Okay, so we've got cells intentionally breaking their DNA, targeted mutations, and this whole idea of untargeted mutagenesis. This is already way more dynamic than I ever imagined.
Right.
But I have a feeling we're just scratching the surface here, right?
You're absolutely right. We haven't even touched on those mobile genetic elements, the jumping genes that are constantly rearranging the genomic landscape.
Jumping genes. That sounds like a whole other level of craziness. I can't wait to hear about that.
Well, that's a story for our next segment. But trust me, it's going to get even more mind-boggling.
Okay, so before we went on that tangent about bacteria playing genetic Jenga, you mentioned jumping genes. What are those all about?
Right, those. They're officially called mobile genetic elements, or MGEs for short.
MGEs.
And they're basically chunks of DNA that can move around within the genome, like little nomads.
So they literally jump around. How does that even work?
Yeah, it's kind of like those cut and paste functions on your computer, Some MGEs, they'll cut themselves out of one spot and insert themselves somewhere else. Others make copies of themselves and then insert those copies so they're more like a copy and paste function.
Wow, okay, so these MGEs, are they just like messing things up as they hop around?
Actually, no. Sometimes they can cause problems, but they're also a key player in this whole read-write genome idea.
Okay.
I mean, think about it. They can carry whole chunks of genetic information with them, like coding sequences for proteins, or even those regulatory elements we talked about earlier, like dimmer switches for genes.
Wait, so as these MGEs are jumping around, they can actually change how genes are expressed?
Yep.
That's incredible. It's like they're rearranging the control panel of the genome.
Exactly. And that's why they're so important for evolution, They add this whole other layer of complexity and flexibility to the genome. They allow cells to adapt to changing environments in ways that wouldn't be possible with just random mutations.
This is blowing my mind wide open. So we started with editing DNA, and now we have these jumping genes rearranging things. How did scientists even figure all this out?
It's a long story, but one of the key players is this amazing scientist, Barbara McClintock. Back in the 1940s and 50s, she was studying maize, corn.
Okay.
And she noticed that certain traits, they were like appearing and disappearing in a way that just couldn't be explained by traditional genetics.
Interesting. So she was like the OG genome hacker, way before anyone else.
Pretty much, yeah. She proposed that these traits were being controlled by what she called controlling elements, which we now know as transposons, which are a type of MGE.
Transposons. Yeah, in the scientific community. Did they, immediately embrace her groundbreaking work?
Actually, no. Her ideas were so radical at the time that a lot of scientists just, dismissed them.
Wow, really?
Yeah, it took decades for her work to be fully recognized and appreciated, but eventually she was awarded the Nobel Prize in Physiology or Medicine in 1983.
That's amazing. And kind of sad that it took so long.
Yeah.
It just shows how revolutionary this whole read-write genome concept really is.
It is.
But, okay, back to these MGEs. You said they can carry coding sequences for proteins. Does that mean they can actually create new proteins?
Yeah, absolutely. Remember we were talking about how cells can shuffle around those protein coding blocks, the exons?
Yeah.
Well, MGEs can play a role in that process too, sometimes even bringing in new exons from other parts of the genome or even other organisms.
Okay, now that is seriously wild. So evolution isn't just waiting for random mutations to create new proteins. It can actually borrow them from other sources. It's like a genetic swap meet.
It is, and it gets even wilder. Sometimes MGEs can even create entirely new genes from scratch through a process called exonization.
Exonization. Okay, break that down for me.
So basically, it's when a non-coding stretch of DNA, which wasn't involved in making proteins, suddenly gets transformed into a protein coding sequence.
Okay.
So imagine you have a bunch of spare parts lying around in your garage, and suddenly you realize you can use them to build a whole new gadget.
Oh, okay. So it's like the genome is full of hidden potential, and
Yeah.
MGEs can like unlock that potential, creating new proteins with new functions.
Exactly. And this isn't just theoretical. Shapiro provides tons of examples of how these processes have happened throughout evolution, leading to the incredible diversity of proteins we see today.
So we've got MGs jumping around, rearranging genes, creating new proteins, and even borrowing genetic material from other sources. It's like the genome is this constantly evolving ecosystem with all these different players interacting and shaping each other.
That's a great way to put it. And it really highlights just how dynamic and interconnected the genome really is, you know? It's not just a static blueprint, it's this constantly changing tapestry woven together by these mobile genetic elements.
Okay, my brain is officially full. I need a moment to process all of this.
I don't blame you, it's a lot to take in. But before you go into overload, there's one more crucial aspect of the read-write genome that we need to discuss. And that's how NGE operates on those larger timescales, over evolutionary time.
Okay, so we've talked about how cells can edit their DNA, how MGEs jump around and rearrange things, and how this all contributes to adaptation. What happens when you zoom out and look at evolution over millions of years?
Well, that's where things get really interesting because
Mhm.
it turns out that NGE isn't just about tweaking existing genes or rearranging small pieces of DNA. It can also lead to these massive genomic changes, like whole genome duplications.
Whoa, hold on. Whole genome duplications.
Oh,
That sounds like a recipe for disaster.
it can be, but it also presents this unique opportunity. You see, with two copies of every gene, cells have more freedom to experiment. One copy can keep doing its original job, while the other is free to mutate and evolve new functions.
So it's like evolution suddenly has a backup copy so it can take more risks and try new things.
Exactly. And WGDs have been linked to some major evolutionary leaps, like the evolution of the vertebrate skeleton.
Wow.
And guess what? Evidence suggests that NGE played a role in facilitating these duplications.
So NGE isn't just about tweaking existing genes or adding new regulatory elements. It's also involved in these massive genomic rearrangements that can lead to whole new branches on the tree of life.
That's right. And it's not just about duplicating individual genes either. Shapiro also talks about how NGE can contribute to the evolution of entire chromosomes.
How?
We're talking about processes like chromosome fusion, fission, inversion, and translocation.
Okay, now you're really getting into the big leagues of genetic change. Chromosome evolution? That sounds intense.
It is. It involves ways that chromosomes can be rearranged, leading to changes in gene order and the formation of new chromosomes.
So NGE is like a master choreographer,
Yeah,
guiding the movements and rearrangements of these massive genetic structures.
that's a great way to put it. And remember, those mobile genetic elements, they're often the ones driving these rearrangements. They hop between chromosomes, carrying genes and regulatory elements with them.
It's amazing to think that these tiny jumping genes can have such a profound impact on the evolution of entire chromosomes. It really challenges the traditional view of slow, gradual change.
It does. And Shapiro argues that these large-scale genomic rearrangements, often facilitated by NGE, have played a crucial role in the diversification of life on Earth.
So from single genes to entire chromosomes, NGE is leaving its mark on every level of genomic organization. This read-write genome perspective is really starting to paint a very different picture of how evolution works.
It really does. It suggests that cells are not just passive recipients of random mutations. They are active players in their own evolution, constantly shaping and reshaping their genomes in response to environmental challenges.
This is all so fascinating. I'm starting to see the genome in a whole new light, not as a static blueprint, but as this dynamic and adaptable system. It's like a living, breathing entity that's constantly evolving and innovating.
And that's exactly the point Shapiro's trying to make. He's arguing that we need to move beyond that old view of the genome as a passive recipient of change and embrace this new understanding of the genome as an active and creative force in evolution.
This has been an incredible journey so far, but I have a feeling there's still more to explore, right? What are some of the broader implications of this read-write genome idea?
That's a great question, and it's one that takes us beyond the realm of pure science and into the world of philosophy and even ethics. Because if cells are truly capable of directing their own evolution, what does that mean for our understanding of life itself?
Okay, now you're really getting me thinking. This is getting deep.
Yeah.
But I think our listeners are ready for that challenge.
I agree, and I think Shapiro would too. After all, he ends his paper with a call to action, urging us to embrace this new understanding of the genome and explore its implications for everything from medicine to agriculture to our place in the universe.
Wow. Okay, I am definitely ready to hear more about that.
Right.
Let's dive into those deeper implications in our next segment. Okay, so we went deep into this read-write genome idea, right? We saw how cells can edit their own DNA, shuffle genes around with those jumping MGEs, even do those massive genomic rearrangements.
Right.
It's totally changed how I see evolution. But you were saying this goes beyond just science.
It does. I mean, if cells are actually like shaping their own evolutionary paths, not just passively reacting to random mutations, that really makes you wonder about the nature of life itself. It kind of challenges that traditional view of a slow Darwinian process and suggests a whole new level of agency that we never really considered before.
Agency. So like instead of just surviving, are you saying organisms are actively creating their future, like little genetic entrepreneurs?
That's a great analogy. Shapiro argues that this cellular agency actually has some profound philosophical implications. It brings up all these questions about the role of chance versus intention in evolution, and it kind of suggests that creativity and problem-solving ability are just, like, inherent in life itself.
Wow, okay, so it's not just about how genes change, it's about how we understand the driving forces behind those changes. That's a pretty big shift in perspective.
Exactly. And it's a shift that has implications beyond just philosophy, too. It touches on things like medicine, agriculture, even our relationship with the natural world.
Let's unpack that. How does this read-write genome idea actually impact those fields?
Well, take medicine, for example. Understanding how cells use NGE could lead to all sorts of new therapies for genetic diseases. Imagine if we could actually harness the power of NGE to correct genetic defects, or even enhance the immune system's ability to fight off infections.
So we could actually turn our cells into these little genetic repair shops fixing problems at the source.
Exactly. And in agriculture, understanding NGE could lead to, more sustainable and resilient crops. Like, imagine being able to engineer plants that can actually adapt to climate change, resist pests, or even thrive in nutrient-poor soils, all without relying on all those harmful pesticides and fertilizers.
So we're not just talking about treating diseases or growing better crops. It's like fundamentally changing how we approach these challenges, working with nature's own ingenuity instead of just trying to control it.
Exactly. And on a broader level, the read-write genome really challenges us to rethink our entire relationship with the natural world. It kind of suggests that life is way more adaptable and resilient than we ever thought, and it really highlights the interconnectedness of all living things.
So instead of seeing ourselves as like separate from nature, are we realizing that we're part of this incredibly complex dynamic web of life, constantly evolving and adapting alongside all the other organisms?
Exactly. And that realization comes with a sense of responsibility. If we really understand the power of the read-write genome, we need to be very careful about how we use that power, both in terms of manipulating our own genomes, but also the genomes of other species.
That's a good point. We can't just go around like messing with genomes without thinking about the potential consequences, both the ones we intend and the ones we don't. It's a powerful tool. It needs to be used carefully and ethically.
It does. And that's where Shapiro's call to action comes in. He argues that we need to have these open and honest conversations about what the read-write genome means. Conversations that involve scientists, ethicists, policymakers, even the public at large.
So this isn't just a scientific discovery, it's a societal challenge.
Yeah.
We need to figure out how to use this knowledge responsibly for the benefit of all life on Earth.
Exactly. And it's a challenge that I think we are totally capable of meeting, if we approach it with some wisdom, with humility, and with a deep respect for the power and complexity of life.
Well, listeners, that's our deep dive into the read-write genome. We've covered so much, from the molecular mechanisms of NGE to its implications for philosophy, ethics, even our future. It's been a mind-bending journey, and I hope you learned a thing or two and maybe even had your curiosity sparked.
Yeah, and remember, this is just the beginning. Genomics is always changing, with new discoveries popping up all the time. So keep exploring, keep asking questions, and keep those assumptions in check.
Thanks for joining us. Until next time, keep your minds open and keep those neurons firing.